Immunodeficiency. What is it? We're going to be looking at that today, and we're going to be looking at a particular disease called HIV and how that can lead to AIDS. So if we look at immunodeficiency, we are looking at someone with an impaired immune system. Their immune system is not working as it should, which leaves them really vulnerable to different diseases. Now, with immunodeficiency, we can have primary immunodeficiency or secondary immunodeficiency. Primary immunodeficiency is from birth. You've got some genetic disorders that are happening and it leaves you super vulnerable to infection because you don't have a working immune system. Secondary immunodeficiency is acquired in your lifetime. So it could be from taking immunosuppressant drugs, or it could be that you've contracted a disease that has wiped out your immune system. An example of a primary immunodeficiency is severe combined immunodeficiency, also known as SCID. So a really famous case of this was of this young boy born in 1971, and his name was David Vetters. They also knew him as the Bubble Boy. David lived in a sterile chamber from birth, and this is why he's known as the Bubble Boy. Everything that went into that chamber, so all his food, water, clothes, toys, it needed to be sterilized before it came in contact with him. He didn't have the right white blood cells. He wasn't producing any immune response that could help him against any normal pathogen, so they needed to keep him safe by keeping him away from exposure of pathogens. A potential treatment for someone with immunodeficiency is a bone marrow transplant. So that is taking donor bone marrow from someone who has working immune cells, giving it to a person with immunodeficiency, and then those stem cells producing a working immune system. In the case of David, he unfortunately did get a bone marrow transplant that was not successful. They didn't screen it very well, and it came from his sister that had a dormant Epstein-Barr virus. Once this virus got into his system, it replicated and it formed a disease that did kill him in the end. So he did die in 1984. Sad end for the Bubble Boy. Secondary immunodeficiency are those that are acquired throughout your lifetime. So it could be that you're taking immunosuppressant drugs, or it could be that you have a disease that's causing your immune system to collapse. So we're going to be focusing on HIV and how it progresses into AIDS when we're looking at secondary immunodeficiency. HIV stands for Human Immunodeficiency Virus, and it is a virus that gets into your cells and it attacks your immune system. HIV affects more than 35 million people worldwide, and we see a high prevalence of it in certain countries, such as sub-Saharan Africa. HIV is a bit of a sneaky disease. In particular, it is going to attack our T helper cells, and this is why it causes such a massive collapse of your immune system. Without your T helper cells, we're not going to be able to activate our B cells and generate that effective third line of defense. So you're essentially left with the first line and second line keeping you healthy. HIV does have a few different stages. So we have the acute infection, which is like the first 2 to 4 weeks after your first exposure to the virus, and you get flu-like symptoms at that stage. After that, we have clinical latency, which is a really chronic condition. It could last for decades even. And this is when we're progressively killing a lot of our T helper cells, and this takes a while. After chronic infection with HIV, our T helper cell count is going to be significantly reduced. So when this happens, we're looking at a number below 200 cells per millimeter cubed, and we are going to develop into our next stage, which is AIDS. AIDS stands for Acquired Immunodeficiency Syndrome, and it's at this stage where your immune system is so compromised that we don't have a working third line of defense, that you're prone to opportunistic infections. So with AIDS, it's not the HIV that kills you, it's more likely that you get a secondary infection like influenza or a cold or something like that kills you, because we don't have defenses against it. HIV is spread through the contact of infected bodily fluids with like basically an opening in your body. So if we do have, let's say, a cut and you introduced some blood of someone with HIV in there, you're introducing the virus into your bloodstream and we're going to have an infection. So we see a lot of HIV cases through shared needles in intravenous drug use. We also have this through unprotected sex, and we have this as well through vertical transmission. So it's the transmission from mother to child during birth. Once HIV gets into our system, it needs to find its target host cell. So our T helper cells are also known as CD4 cells because they have this receptor called CD4 on its surface, and that lets our HIV in. HIV has this protein on its surface called GP120, and that is complementary to the CD4 receptor. When these two things bind together, our HIV docks into our T helper cell, enters the T helper cell, and it will start to replicate. HIV has RNA inside of the viral particle instead of DNA, so we call this a retrovirus. We also see this in influenza. So when it gets into our cells, we first need a particular enzyme that the HIV will have called reverse transcriptase, and it will convert the RNA into DNA. When this happens, we then insert this DNA into our host DNA genome. So we have this enzyme called integrase, also produced by the virus, that is going to basically snip a little bit of our DNA, insert that DNA in, and it's going to be read like any other DNA in your body. Once that viral genome is in our own DNA, our cells hijacked. We are going to be making lots and lots and lots of this virus in our cells, where it's going to be lysed out later on, and it'll infect more and more T helper cells. So if we have one T helper cell in our lymph node that is infected, it's going to burst out and infect all the other T cells that we have hanging out in our lymph node. Now, there are a small number of people who are naturally immune to HIV because their T helper cells don't produce the CD4 receptor. Like, if you're not sure if that's you though, don't go testing that out. Like, please don't. There are no treatments which will completely reverse the infection of HIV. So any infected cells that you have in your system, they're gone, they're dead. We can't do anything about them. What we can do is slow the progression of the disease. So we can slow how the virus spreads from cell to cell within the host. We are looking at a particular class of drugs called antiretrovirals, and these are an example of rational drug design. So if you remember rational drug design when we looked at Relenza, it is knowing a little bit about our target molecule and blocking the action of that target molecule. In this case, we are blocking the action of the enzyme that helps the retrovirus to actually take over the host cell. So we can target integrase, or we can target reverse transcriptase, or any other machinery we have in that virus. Once these are targeted, that virus is not able to replicate as it should, and it really does slow the progression of the disease to the point where your viral load might be close to zero. That means that your number of viral particles is pretty much non-existent in your body. There are financial and logistical barriers that stop us from getting these antiretroviral drugs to the countries that really need it. So if you look at this drug, the cost of it varies so much from country to country. In the US, the treatment for this will cost you about $3,000 a month. And in Australia, where we have, a pharmaceutical benefit scheme that subsidizes the cost of our life-giving drugs, this will cost you about $40 a month. So massive amounts of difference in cost. So right now it's not really widely used in areas like sub-Saharan Africa. Because of the difficulties in getting antiretroviral drugs to those that do need this treatment, the best defense against HIV is education. So we're looking at different ways to educate the population so that they can prevent transmission of the disease. Lots of research has been done into more treatments for immunodeficiency. So we're looking at things like CRISPR and DNA manipulation. Who knows what the future of treatments is going to look like, but overall research is happening, scientists are learning a lot, and science is awesome.